Becky's a PhD candidate in the De Horta group, as was Reese, who you heard from just a few talks back. So the De Horta group is, of course, headed by Dr. Nathalie De Horta, and they look at neuronal development. So Becky's focus is on a little bit what you might call the other side of development, the process of aging. So Becky's work looks at what goes wrong when neurodegenerative processes occur in later life, and in particular in the context of Parkinson's disease. So today, Becky is giving us a talk called When Neurons Die, an overview of common neurodegenerative disorders. And yeah, I'm really looking forward to this one. Take it away, Becky. Thank you.
Okay. So I'll just set this up. I think it might be. Can you see that okay? Alrighty. Thanks. All right, hello everyone. First of all, I just want to say a massive thank you to all of you for coming today and for the organisers. This has been a pretty incredible day. I've already learned quite a bit, and I really feel honoured to be able to present with such incredible speakers that we've had. So, as Angela said, I'm going to talk about neuronal death today and the neurodegenerative disorders that are quite prevalent in society. So, the brain's a pretty incredible, complex organism, but it's very powerful in controlling our behavior, our cognition, how we move, and everything. What happens then when these neurons that we've looked at today die? So the basic units of our bodies come down to these microscopic cells, which have regular life cycles, just like we do. And just like we are born like cells, like Reese explained earlier, and then we grow and we die. Now, most cells in our bodies reproduce at a non-stop, constant rate. If you're injured, your skin will make new skin cells to heal the wound. If you lose blood, your body will generate more blood cells. However, neurons are not quite that simple. They're the longest living cells in the body, and when they die as an adult, it is far, far more difficult for new neurons to develop in their place. But why would a neuron die? One very common mechanism is necrosis, which is the accidental and uncontrollable cell death due to external environmental influence. So it could be like infiltration of pathogens, a blow to the head, traumatic injury that causes cellular damage. These cells will swell up, rupture, and provoke an inflammatory response, like Rick and Nadia were talking about. Another mechanism is apoptosis, and I noticed Reese touched on this a little bit, but apoptosis is defined as genetically programmed cell death, where the cell actively kills itself, a cell suicide, if you will. This is responsible for providing homeostatic balance in order to control cell populations, but it also prevents malfunctioning cells to trigger further damage. When the apoptosis function doesn't work, cells may divide uncontrollably, and this seeds dangerous mutations to other cells. This is ultimately how tumors are created and subsequently cancer. Now, sometimes these pathways are involved in rapid and unnatural depletion of neurons, which can cause serious chronic diseases. These are called neurodegenerative disorders. Some include dementia, Parkinson's disease, Huntington's disease, ALS or Prion's disease, like Creutzfeldt-Jakob's disorder. And you may have heard of some of these, and sadly you may know someone diagnosed. And this wouldn't be that surprising because, in fact, neurodegenerative diseases affect millions of people, with the most common being Alzheimer's, affecting 50 million people worldwide. But despite their prevalence, there are currently no neurodegenerative diseases that are curable. It should be noted that these diseases may not be triggered by a change in neuron number alone. Other factors, such as degeneration or death of cells, may include changes in the connections between these neurons and the way in which they communicate. So as Brian discussed much earlier on in the day, neurons receive messages from other neurons, transmitting this information as an electrical impulse down their long extensions to stimulate the release of chemicals or neurotransmitters, which are transported to the receptors of the receiving cell. In neurodegenerative diseases, these neurotransmitters are depleted, inhibiting cell communication between cells throughout and progressing on to cause further problems in different pathways and in regions of the brain. So you can kind of see here some neurotransmitter examples in these diseases include dopamine, acetylcholine, gamma-aminobutyric acid or GABA, and glutamate. Now, within these neurons, there are cell bodies that contain their genetic code, like DNA, as well as factories that produce energy and things called proteins. Proteins may include things like enzymes that catalyze reactions, but are also very important for maintaining a scaffold of the neuron, transporting molecules and facilitating chemical reactions. Neurodegenerative diseases are often massively linked to specific proteins that misbehave and sometimes become toxic. Some examples of these include beta amyloid or tau in Alzheimer's, alpha-synuclein in Parkinson's, and as luck would have it, Huntington's in Huntington's disorder, and prions in prion diseases. Now, a native protein is code-wired to have a set folded configuration to allow it to function normally. When this folding goes awry, misfolds can aggregate and accumulate into bundles, forming oligomers and fibrils. Now, these get deposited within the neurons that we've looked at, and these aberrant proteins place massive stress on the neurons due to losing normal function, but also by rendering deadly events such as increasing reactive oxygen species that Rick was talking about, dysfunction of chemical release that we saw before, impairing protein degradation, or the systems of other factories within the neurons, DNA damage, and also inflammation and immune response. And the death of these neurons alone can trigger inflammatory and immune response, which may further exacerbate neurodegeneration. Now, as I briefly mentioned, these aggregated forms of proteins can cause neurodegeneration by intracellular damage to different parts of the cell. One factory of the cell that you may remember from high school biology as the powerhouse of the cell is referred as the mitochondria. Neurons require a great deal of energy to power mitochondria, so healthy mitochondria is essential for this neuronal functioning. And oxidative stress, as you can see here, or certain gene mutations can actually cause damage to the mitochondria. And when mitochondria are detected as defective, they undergo mitophagy, whereby ubiquitin enzymes help to destroy the membrane proteins and recruit other factories called a phagosome, which digest the mitochondria, leading to its degradation. This prevents mitochondrial oxidative stress and toxin susceptibility from affecting the rest of the cell. However, when mitophagy is inhibited, the mitochondria can no longer regulate itself and can damage the rest of the neuron. This is commonly found to be linked with neuronal degeneration, predominantly Parkinson's, but in some cases has been identified in ALS, Alzheimer's, Huntington's disease, as a result of these pathogenic proteins or from genetic mutations involved in the process. So let's look at some of these mechanisms occurring in these common disorders. Alzheimer's is the most common neurodegenerative disorder. It is steadily increasing in prevalence, with an estimated 150 million people expecting to be diagnosed by 2050. And Alzheimer's is characterized by a gradual and progressive decline in cognitive function. It initially presents with memory impairment and then can further cause cognitive and executive functioning that leads to poor judgment, difficulty with simple tasks, loss of direction, navigational information, as well as changes in mood and personality, say depression or irritability or aggression. Now, the hippocampus is responsible for memory, whether that be spatial memory or memory of facts and events. Therefore, this most prominent clinical symptom, memory loss, is correlated with neurodegeneration in the hippocampus. The endurance of neuronal loss is correlated with the progression of the disease. And as the disease advances, the frontal cortex and other cortical, subcortical regions also experience neuronal losses that sometimes can be very severe. This is often when impairment in social behavior, language, and reasoning develops. So PET scans and autopsies display a severe shrinkage, as you can see here, of the hippocampus, atrophying of the cerebral cortex, and also significant enlarging of these ventricles that I think Nathan might have mentioned that carry the cerebrospinal fluid. Okay. The pathogenic proteins of Alzheimer's disease include amyloid and tau proteins. Amyloid precursor protein helps with the growth of neurons, but it has one major problem. It contains a dangerous part that has a higher susceptibility of forming these plaques and aggregates. And this part often gets cut straight through the middle, releasing two harmless fragments. However, sometimes this protein is cut at different parts and kind of releasing the dangerous intact fragment called A beta fragment. And this problem lies once this happens to many amyloid precursor proteins, causing these amyloid beta fragments to aggregate together, form bundles that create fibrils, and then progress into amyloid plaques. The other, tau protein, under normal conditions, stabilize these structures that are necessary for maintaining the neuron's shape and transporting vesicles, like the synaptic activity that Brian was talking about. These contain useful molecules that further onto the extremities of the neuron. And these are called microtubules. In Alzheimer's, the tau protein is modified and aggregated like amyloid beta and detaches from the microtubule, damaging the neuron. Now, this image here shows how both variants, with tau causing neurofibrillary tangles occurring inside the cell, and also amyloid precursor proteins that create amyloid beta plaques in the extracellular space. So coming in as the second most prevalent neurodegenerative disease, where my research is as well, Parkinson's. Parkinson's affects 10 million people worldwide, with 38 more Australians being diagnosed every single day. The disease is typically known for its symptom progression of inhibited motor functioning, and this poor movement control is often expressed by postural instability, resting tremors, as well as fixing and slumped posture. And these can prevent normal locomotion, coordination, and gait. And despite these typical presentations of symptoms, Parkinson's is not just the shakes. It can also cause difficulties swallowing, smelling, chewing, and speaking, as well as presenting with gastrointestinal dysfunction, like hiccups, constipation. And in fact, some of these symptoms actually occur earlier on in the disease before tremors even develop. Additionally, there are many other associated diseases and symptoms, such as emotional instability and stress in the form of depression and anxiety, or inhibited cognition such as reduced memory, attention, executive functioning, amongst many, many others. Now, these symptoms are caused by a depletion of dopamine, one of these neurotransmitters that we were talking about in the brain, specifically the reduction of dopamine from dopaminergic neurons within the substantia nigra. You can kind of see a little bit here how it actually gets less dark in a Parkinson's brain. Now, these neurons principally innervate the basal ganglia, and this is responsible for movement and gait control. And it is not until actually 80% of dopaminergic neurons are depleted within a certain part of this basal ganglia, the caudate putamen, do we actually see presentations of the symptoms occurring. And as there is still no known cause and very few biomarkers characterizing the disease, clinical presentations are the standard method of diagnosis. So Parkinson's sufferers are often not diagnosed until they are in very, very late stages of the disease, sadly. Now, the last disease that I want to touch on today is purely based off my own selfish interest. It's the rather rare but fascinating Prion's disease. So Prion's disease is caused by the abnormal proteins that we talked about before that build up in cells, but they actually seed aggregation into neighboring proteins that were once functioning normally. Over time, the brain actually slowly shrinks, causing tissues to form holes like a sponge. So this is one example of Creutzfeldt-Jakob's disorder. And because of this, the disease is extremely contagious and actually transmissible. And the first case was denoted the term kuru and developed in Papua New Guinea as a result of others eating each other with infected with the disease or with these prion proteins. Prion's disease symptoms are quite severe in that they include memory impairment, personality changes, as well as difficulties with movement. And they can be very, very deadly, with survival time usually lasting a few months. Interestingly, however, the mechanism in which prions work have recently been shown in other neurodegenerative disorders. This kind of emphasizes the importance of knowing how this disease works and how to stop it from progressing to transfer this to other diseases like Parkinson's. So there are a multitude of factors that can increase susceptibility of developing neurodegenerative diseases. It could be mutations in genes associated with some of these diseases could play a role in the mitochondrial or cell factory functioning, or are affected by epigenetics, the reversible genetic changes that can occur from the environment. Age, of course, is naturally associated with an increasing decline of mitochondrial function. We see a majority of these cases, like Alzheimer's and Parkinson's, occur over the ages of 60. And some environmental toxins, such as pesticides and herbicides, and even smoking in some cases, can target the mitochondria and generate oxidative stress. As cell death can be amplified through autoimmunity and inflammation, people's individual and variable different levels in immunity can also increase or change their susceptibility of an immune response developing. As well, we see sex and endocrine levels also implicated in neurodegenerative disorders. So 2/3 of Alzheimer's cases occur in women, and men are 1.5 times more likely to get Parkinson's than women. Now, additionally, blows to the brain or damage caused by a stroke can cause the necrosis of these cells, like you mentioned before, but also slowly starve them of oxygen and the nutrients that they need to survive. So some theorized strategies to avoid developing a neurodegenerative disease. I know a lot of people really interested in this sort of thing, these preventative strategies, many of which include getting adequate sleep and exercise, being intellectually and cognitively stimulated and socially active, but also avoiding toxins. Now, I know a healthy diet is another contributing factor, but don't be careful with what you read, because there's a lot of conflicting theories about what is actually beneficial. Antioxidants are usually your safe bet, but as long as you kind of remain that healthy diet, vegetables, those sorts of things, you can increase your general health in the long term regardless. Now, while there is still no known cure, there are currently many palliative treatment available to control the symptoms and manage everyday tasks. For example, we have different types of therapy like physiotherapy, speech pathology, psychiatry appointments, and then you can also get drugs that target the neurotransmitter levels through synapse activity, like Brian was talking about. Levodopa is one in Parkinson's that targets the precursor of dopamine, kind of allowing it to synthesize and increase in the brain. Riluzole in ALS blocks the release of glutamate as like an inhibitor. And cholinesterase inhibitors prevent the actual breakdown of acetylcholine that's depleted in Alzheimer's disease. We also see anti-inflammatory drugs like NSAIDs in Alzheimer's to kind of reduce this inflammation and for further oxidative stress, as well as antipsychotic drugs for psychological and behavioral symptoms, and analgesics to control pain or allow pain relief. The prevention of abnormal protein aggregation is an interesting one. It's something that's looked into a lot, still not really effective yet, especially as a curative agent, but it can maybe cause in future the targeting of misfolded proteins to allow their degradation, or even just inhibiting this aggregation or these fibril formations. Other strategies, some are surgical interventions, such as deep brain stimulation, which is the most common actual surgical procedure used to treat Parkinson's and sometimes in Alzheimer's, but it uniquely doesn't destroy cells or damage tissue. Electrodes are placed in the hemispheres and linked to this battery-operated device. You can see here. And it's underneath the skin below the collarbone. And the device delivers continuous electrical pulses that transmitted through electrodes to the brain. And these pulses interrupt that irregular electrical signaling that's caused by the disease, regulating the signal patterns back to normal. And an interesting therapeutic strategy is stem cell regeneration. It's still currently and vastly in the works and being researched today. And it's a quite powerful strategy for treating neurodegenerative disorders, as it starts with stem cells that are undifferentiated, early developed cells, like you might have seen Reese talk about in early stages. And you can take these from adult tissue like the skin, embryos, which has got ESCs here for embryonic stem cells, or even reprogram for differentiation called induced pluripotent stem cells to be used to replace the damaged or lost neurons, glial cells, like we see in these neurodegenerative diseases. So while we have a better understanding of how many of these processes occur, there is a lot more research to be done. We are still ways away before finding a cure, and with the continual increasing prevalence, every person is at risk of developing a neurodegenerative disorder, or at least knowing someone who sadly will. And I hope you agree with me when I say the brain is definitely worth protecting. So again, thank you so much to everyone, the organizers. Today has been pretty awesome. Does anyone have any questions? I will just preface, my main research focus is Parkinson's disease. So I will do my best to answer other neurodegenerative disorder questions too. Cool, thanks.
Awesome, thank you, Becky. That was very interesting and very informative. And I had a lot of questions as you were going, but you sort of answered one after the other. So it was, yeah, very well laid out, and yeah, really great talk. Thank you.
Yes.
So there's a question here from Zana, and they're asking, or they're noting, please give some information about the pattern of methylation in neurodegenerative diseases.
Are you referring to, I'm assuming she's referring to DNA methylation, like you can see in epigenetics? Yeah, so I'm definitely not a geneticist, but I do know that there are some sort of risk factors that link, especially as we see a lot of genetic derivations of neurodegenerative cell diseases, as well as these environmental causes. So sometimes environment plays an impact on actually kind of damaging cell DNA and triggering these sorts of onsets, like the methylation, which can be the actual changing of the code to be methylated. Essentially, I'm kind of butchering this, but to then change the encoding proteins that develop as an onset. Does that make sense? Oh, there's another one.
Yeah, another question from Jen. There's a few popping up now, which is great. So Jen has asked, how strong is the genetic link as a precursor in Parkinson's? Is it familial?
Yeah, that's a great question. I'm really pleased that you're asking your Parkinson's question. There's actually only 10% or 15% of cases that are inherited or hereditary in Parkinson's. So not many, and yet all research is conducted just with genetic or toxic models. We do see probably more the case of like an interplay between genetics and environmental influences, but there's a lot that's unknown, and there is no known cause either, as well as a cure for Parkinson's. So a lot more to be done. I can list off some of the mutations and those sorts of things that's involved specifically in Parkinson's, but that might not be too interesting.
Yeah, thank you. That's a very good answer. And there's one more from Hannah, and she asks, how do you weigh up the side effects of drugs such as levodopa versus their effectiveness as a treatment for Parkinson's disease?
Yeah. There's a lot of actually drugs available. Some that synthesize these dopaminergic precursors. Some that allow a better uptake of dopamine, and there's some that combine those two. There's a bunch of different variations, but unfortunately, they're still more palliative than a cure. They're fixing something that's already damaged or something that's more innate that we don't know the cause of. So while they are really effective in being able to kind of regulate symptoms, in some cases, they can't really do anything for the long term. But of course, things like even levodopa, you actually get things like side effects of worsening as almost like a tolerance developing from the drug as well, where people can actually, in some cases, their symptoms worsen. So there is still a lot more to be done there.
That sounds like it really is a matter of, as the sort of question alluded to weighing up the pros and cons for every individual and sort of tailoring their treatment.
Exactly.
Considering both sides, the possible benefits and the possible side effects.
Yeah.
Alex also asked a question, and he has said, is there advances of using viral vectors in delivering correct genes in curing neurodegenerative disease?
Certainly, yeah, there certainly is. I'm trying to think of some that are good examples. They're usually, I think, related to like the proteins, but I can't think of any names at the moment. There are, and they are also being worked on. I don't know how effective they've been as of yet, but we can even use viral vectors as models in the lab as well, and we can determine this. So I think that's something as well I want to pursue in my research. So watch this space, Felix.
Sounds like a very good question.
Yeah, it was good
relevant
question.
to your work.
Yeah.
There was a comment which I meant to mention as well from William. While you were doing your talk, he said, this presentation has the best memes so far. So thank you very much for that.
Sorry, I hope it wasn't too
No, very good. And very, like I mentioned before, very interesting and very informative. So thank you so much for your contribution today.
Thanks, Andrew.